almost 70% chapter is covered. Okay, just remaining portion is there. One remaining portion is there in this chapter, that is nothing but only the applications of biotechnology. So now we will see the applications of biotechnology in detail. But before we start the application of biotechnology, first just a quick revision on the previous lecture, that is nothing but the rDNA technology. Okay, so what was studied in rDNA technology then? rDNA means it is recombinant DNA technology. There are several steps. How many steps are there in rDNA technology? There are six steps in rDNA technology. First, isolation of a DNA fragment from donor cell. That is the first step. Then second, insertion of that fragmented part into the suitable vector. Okay, that is the second step. Third step, it is nothing but the combination of that, means that rDNA or foreign DNA is again inserted into the host cell. Which host cell then? That host cell, it is nothing but it is the bacteria, that is Escherichia coli. That is the third step. Now what happened in fourth step? In the fourth step of rDNA technology, the process of transformation takes place. Okay, means there is a formation of multiple copies of transformed cells, host transformed cells. Okay, that is the fourth step. Now what happened in the fifth step? The multiplication of transformed host cell takes place. After that, the last step, that is nothing but the extraction or formation of suitable product from that cells. These are nothing but the steps which we already studied in our last lecture. That is nothing but the processes of rDNA technology. Okay, and see, in this whole chapter, PCR and rDNA technologies, basically the steps as well as diagrams, both are important regarding PCR and rDNA technology. And suppose if the short nahi long question arises, okay, so for sure it is from PCR and rDNA technology. Apart from that, the 2 marks question or very short questions arises on the applications of biotechnology. But as we already started the application of biotechnology in medicine, okay, in medicine. So in this medicine, the first application given is in the form of insulin. The first application given in the form of insulin. So this insulin is also very important regarding your exam point of view, because many times the question asks on the structure of insulin, how insulin is prepared and all. Okay, so finally we are ready to start our fresh lecture, that is nothing but the application of biotechnology in the stream or in the medicinal or in the medicine way. Okay, so the first example given in your textbook, it is nothing but the insulin, and second, it is nothing but it is vaccine. These two examples are given as a application of biotechnology in a medicine, that is insulin and vaccine, and both are having very important regarding exam point of view as well as regarding Neet point of view. Many MCQs ask from this section. So we will start in detail what is insulin, how it is prepared, and apart from that, how artificial insulin is prepared in the laboratory. Okay, so first of all, what is insulin? Insulin, it is nothing but it is the, what is insulin? Tell me, anyone know what is insulin? What is insulin? General concept. What is insulin? Sada, text me. What is insulin? No idea. Anyone is having idea? See, there are say 5 students which are online now. Okay. So what is insulin? Anyone having idea? What is insulin? No idea? Okay. We can carry
The
on.
function of insulin is control the sugar in a blood.
Yeah, that is the function. But suppose someone asking you what is the definition or what is insulin, then what should be your answer? So, okay, don't worry. I am going to telling you that. So, insulin, it is nothing but it is one of the peptide hormone. What is insulin? Suppose anyone asks you what is insulin, then insulin, it is nothing but it is the peptide hormone. What is insulin? It is nothing but one of the peptide hormones. Okay, what is insulin? Insulin is nothing but one of the peptide hormone which is having the function to regulate the sugar level within the blood. That is nothing but the definition, or we can say characterization of insulin or the function of insulin. What is the function of insulin then? Insulin just maintain the sugar level within the blood. That is the function of insulin. And what is the definition of insulin then? Insulin is nothing but it is the peptide hormone secreted by certain cells of the body. Now which cell secreted this insulin? You know, pancreas. Okay, our pancreas. Okay, so where insulin is synthesized within our body, the pancreatic cell. But pancreatic cell, which pancreatic cell? Then the beta cells of Langerhans. Okay, so which cell synthesize? See, pancreas is having large number of cell. It may be alpha cell, beta cell, gamma cell, and all. But particularly, which pancreatic cell synthesize the insulin molecule? Then it is nothing but the beta cells of Langerhans. Which cell? It is beta cells of Langerhans of pancreas secrete the insulin, and it is normally synthesized in your body. Okay, because this is the normal metabolism of synthesis of insulin. So hopefully you have clear regarding what is insulin, where it is synthesized, and what is the function of that insulin. Okay, just insulin, it is the first point regarding insulin. Insulin, it is nothing but the peptide hormone which is synthesized by the pancreatic gland or pancreas. Okay, see, then pancreas मध्ये कुठले cell? Then beta cells of Langerhans play an important role to synthesize this insulin. Okay, now we will see the structure of insulin, general structure of insulin, and after that we are going to study how artificially this insulin is synthesized within our body. Okay, so this is regarding the introductory part of insulin. Now the insulin, it is made up of two chains. The insulin, it's a molecule, okay, which is made up of two chains. Okay, so alpha chain, A chain म्हणू शकता, or we can say alpha chain and beta chain. Okay, alpha chain and beta chain. So how many molecules, how many chains are present in the insulin molecule? So the insulin molecule, it is having the presence of two chain, alpha chain, or you can say A chain. A na म्हणता alphaच म्हणायचं. Okay, so first it is alpha chain and second it is beta chain. Now this another point, the alpha chain, it is made up of 21 amino acids. How many amino acids are present in alpha chain? The alpha chain, it is having the presence of 21 amino acids, and the beta chain, it is having the presence of 30 amino acids. Okay, this is very important. See, structure विचारू शकतात regarding exam point of view. So you have to keep it in mind that the insulin molecule, it is having made up of two chain, alpha chain and beta chain. First alpha chain, it is having the presence of 21 amino acids. Second, it is beta chain, it is having the presence of 30 amino acids. Okay, now see the diagram. For example, this is alpha chain, this is the short chain. Okay, this is alpha chain and this is beta chain. Okay, for example, this is alpha chain and this is beta chain. So this insulin molecule, first of all, present inactively in the form of proinsulin. Okay, in the form of proinsulin means these insulin molecule, is present within our body in the form of inactive form. But when the C chain is present, means when see this is alpha chain, okay, and this is beta chain. So if there is a presence of C chain along with alpha chain, for example, see, I'm using different pen. So see, for example, this is nothing but the C chain. This is nothing but the C chain. This is nothing but the C chain. असं पण म्हणू शकता. Okay, so this is nothing but the C chain which attaches alpha chain and beta chain. Okay, the dotted blue color line which you are showing in this diagram, it is nothing but it is the C chain. And when this C chain is present in between alpha and beta chain, then that insulin, it is called as proinsulin, and this proinsulin, it is nothing but the inactivated form of insulin. Now what is the meaning of inactivated form of insulin? Inactivated form of insulin means the insulin which can't function properly, which can't function properly means which can't control the sugar level, or which can't maintain the sugar level within the body. That is nothing but the proinsulin. Now, how this proinsulin become insulin then? How this proinsulin become insulin? Okay, so what happened? See, alpha chain and beta chains, both chains are separated. Okay, so for the separation, for the joining of these two chains, that is alpha chain and beta chain, the C chain helps. Means the C chain in between alpha and beta chain helps to bind the alpha and beta chain. Now, how this C chain helps to bind that alpha and beta chain? So, see, there are several sulfide bonds which are present on both alpha chain as well as beta chain. Okay, so that disulfide or sulfide bonds get attaches from one to another. That sulfide or disulfide bonds get attached from one another, okay, through the C chain. And once alpha chain and beta chains are get attached by the disulfide bond, then C chain gets disappeared. Then after that binding, the C chain get disappeared. Okay, so this C chain get disappeared, and in this way the pre-insulin is converted finally into the insulin, and this insulin, it is having the functional one. Okay, and this insulin is the functional one. So in this way, this is the normal structure of insulin which is present in your human body. Hopefully you all understood regarding the structure. See, अशी पण diagram काढता येते, or in a long chain also there is one diagram. Okay, means अशी पण काढली diagram तरी it is okay. Then or this diagram, that is, for example, this is A chain. Okay, then this is B chain, and in between this there is a presence of this C chain. Okay, so this is also the diagram like DNA fragment, and this is also the diagram, but just to show that disulfide bond and that C-shaped structure of C chain, because the name itself indicates C chain. Okay, so because of that only this diagram is considered, that is having the intermediate C-shaped structure. So this is regarding the normal structure of insulin which is present in our body. Okay, so there are some scientists which propose this model or which propose this structure of insulin, and that that name of scientist, it is Edward Shapper. The name of that scientist, it is Edward Schaffer. Okay, the name of scientist, it is Edward Schaffer in the year 1916. In the year 1916, and the name of the scientist, it is Edward Schaffer. Okay, and but this is not the end. The studies going on the insulin. Okay, means this is not the complete one. After the study is also going on. So Later on, the another scientist, the name of the scientist it is Hakura. Okay, the name of the scientist it is Hakura. Okay, in the year 1977. See, I already told you this insulin it is having the presence of two chain, that is alpha and beta chain. So that complete structure having the presence of chemically alpha and beta chain with the 21 amino acids on alpha chain and 30 amino acids on beta chain. This scientist, that is Hakura, in the year 1977 proposed the confirmed structure of insulin. Okay, now this is regarding the normal conversation of insulin. Now, how this insulin is formed artificially by the use of rDNA technology. By the use of rDNA technology, how this process of insulin means production, how this production of insulin is takes place within the laboratory by using rDNA technology, that is important and that we are going to study now. Okay, so see, you know, in normal structure of insulin there is a presence of two chain, alpha and beta chain. Okay, so likewise when you are producing insulin within the laboratory, first of all you have to made or you have to form that two chain artificially. Okay, so how that happens? So see, in the production of insulin within the laboratory by using rDNA technology, first and important point is there is a formation of cDNA. There is a formation of cDNA. cDNA means it is complementary DNA. As I already told in last lecture, in our last lecture, what is cDNA? It is nothing but it is complementary DNA, and where it is present, this cDNA is always present in the eukaryotic cell. Okay, so first of all, the cDNA form. But how the cDNA form? I told you that processes, that is transcription as well as translation. What happened in transcription? From fragment of DNA, there is a formation of mRNA molecule. This is nothing but the process of transcription. Okay. This is nothing but the process of transcription. Okay. So what happened? For the formation of this complementary DNA, this mRNA is again converted into the DNA molecule by the enzyme reverse transcriptase. Okay, so by the reverse process of transcription and by the help of enzyme reverse transcriptase, this mRNA molecule is again converted into DNA, but with carrying some specific sequences or genetic material, and that DNA it is called as cDNA. Okay, and from that cDNA we are going to prepare the artificial insulin within the laboratory. So what happened in the first step? C, from that cDNA. Okay, so we are preparing two chains now, alpha chain and beta chain. So what happened? First we are taking the fragment of the cDNA molecule, then this purna cDNA molecule. Okay, this is cDNA molecule, and this is, for example, some vector. Which vector it is used in rDNA technology? It is nothing but the PBR322. Okay, rapidly used vector kutla hai? It is PBR322. It is the rapidly used vector. So first we are going to see how alpha chain is formed and then how beta chain is formed. Okay, so from this cDNA we are getting only the fragmented portion. Okay, now this vector, but one part is cut. Okay, now what happened after that? The fusion is formed, and that is nothing but our alpha chain. This is nothing but our alpha chain. Okay, so this is the formation of alpha chain within the laboratory by using cDNA. Just what happened? From that cDNA, we are getting only the one fragment. Okay, that one fragment of DNA is then mixed with plasmid, okay, that is PBR322. Then both this cDNA and PBN, we don't require whole DNA and whole plasmid, whole DNA molecule. We need only the fragment portion of DNA molecule. So we are taking that fragmented portion of DNA molecule, okay, then that fragmented portion of DNA molecule get incorporated into the plasmid, okay, and in the last step there is a insertion of cDNA within the plasmid, okay. So in this way the alpha chain is formed within the laboratory. Now likewise alpha chain, the beta chain is also formed. So what happened then? See, after alpha chain, another, it is also like that. Only the difference is in the alpha chain, the cDNA which we extracted, it is having the constituents of how many amino acid? It is having the constituents of 21 amino acids. But in beta chain, it is having the constitution of 30 amino acids. That is only the difference. So likewise, both these steps, okay, the final products we are getting, it is nothing but it is beta chain. It is having the presence of beta chain. So this is nothing but our beta chain. So this is our beta chain. Okay. So now what happened in the next step? Only we are generating the alpha chain and beta chain within the laboratory. Now what happened? After producing alpha chain and beta chain, these both the chain are mixed in suitable host cell. That is nothing but the E. coli. Okay. So after that, both that chains, okay, that is alpha and beta chain, which is having the presence of cDNA fragment, both are mixed in a suitable host, that is nothing but the bacterial cell, okay. And after that, see, this is alpha and this is beta, and this is the presence of DNA fragment, and it is nothing but the E. coli. Okay, so in this way we are producing insulin within the laboratory by means of rDNA technology. Up to this, first of all, असं कोणी तुम्हाला विचारणार नाही how the process of rDNA technology is actually carried out. Okay, this is not the question. Question arises on this insulin. It is that what is the structure of insulin? Just you have to draw that alpha chain, beta chain, then how it is connected and all. But for your knowledge, okay, I am explaining how these steps are carried out in the laboratory by the uses of rDNA technology. So once the bacteria, that is host cell, accept that both the chain, alpha chain and insulin, alpha and beta chain of the insulin, then it is called as prepro insulin. It is called as preproinsulin, okay. And after that preproinsulin is formed, that converted into proinsulin, and that proinsulin is finally converted into insulin. So there are steps. First, there is a formation of preproinsulin. That preproinsulin is converted into preinsulin, and that preinsulin is converted into insulin. Okay, so this is the method of formation of method of formation of artificial insulin by means of rDNA technology. Okay, so that method, okay, and that chains which are accepted by the host cells, and there is a formation of artificial insulin within the laboratory. So the name of insulin which is prepared in the laboratory, okay, the first insulin which is artificial insulin and which is prepared in the laboratory by means of or by using rDNA technology, it is nothing but the humulin. What is the name of that insulin? Always remember for Neet concept, okay? Humulin, it is nothing but the first artificial insulin which is produced in the laboratory by using rDNA technology, okay? And this humulin insulin is made up in the year 1983. In the year 1983, and one of the American industry, that is Eli Lilly. one of the American industry in the sense it is one of the chemical industry. Okay, so Eli Lilly, it is the name of an American industry who discovered this humulin in the year 1983, and afterwards in India also it is made. So this is regarding your detailed summary of insulin, and in this way your first medicine application in the process of biotechnology is finished. That is nothing but the insulin. So see, one term is mentioned in your book, that is the genes are inserted by the site of beta galactosidase gene of the plasmid. The recombinant plasmids were separated, transferred into E. coli host cell. The host produced. ओके, मी तेच सांगितलं आता तुम्हाला जे त्यांनी सांगितलं आहे. फक्त काय आहे त्यांचं म्हणणं, the pre-pro insulin converted into pro insulin, then the pro insulin is converted into insulin. So for that step there is a requirement of certain agent, and that is nothing but one treatment, that is called as TRPC treatment. That's it. And one word is also mentioned, that is beta galactosidase. So beta galactosidase, it is nothing but one of the enzyme which is responsible to remove that C chain and connect that alpha and beta chain within the laboratory naturally. Okay, so this is regarding all summary of insulin. Now the next concept or next medicinal important given, it is nothing but it is vaccine. It is nothing but it is vaccine. Now how these vaccines are prepared and what is the meaning of vaccine? So vaccine, in general definition of vaccine, so vaccine, it is nothing but the antigenic preparation of microorganism. Okay, word itself indicate antigenic. See, antigenic preparation of microorganism. Antigenic preparation of microorganism means what? For example, there are several vaccines which are recently present. Okay, for example, you know, there is a polio vaccine. Okay, polio, this is one of the disease. Okay, but luckily now this disease is completely disappeared from the earth, as the full population is get vaccinated by this polio vaccine. Okay, so about 1% or very rarely persons are still shown that they are having the polio, but not more than that, because up to 2023 there is मोदीचा एक प्लान आहे की after 2023 there is no patient seen by suffering from polio. Okay, so I am just telling you the examples of vaccine. First it is polio vaccine, then second it is hepatitis vaccine. Okay, that is HBV hepatitis vaccine. Then apart from that there are several vaccines. For example, recently to fight against corona there is a vaccine. Okay, so that is again the vaccine, that is corona vaccine. So this is nothing, Covishield, Covaxins are nothing but the examples of vaccines which fight against the coronal infection. So now what is vaccine? मी just तुम्हाला वॅक्सिनचं ओव्हरऑल समरी सांगितली आणि एक्झाम्पल सांगितले. Now the detailed study of vaccine, how this vaccine is prepared. See, in your book only the structures are given. Okay, not detailed these procedures are given, but for your Neet point of view the detailed concept is given in your NCERT. Okay, so what is vaccine? Vaccine, it is nothing but the antigenic preparation of microorganisms. Okay, now vaccines provide the active acquired immunity. Now what is mean by active acquired immunity? For example, जर तुमचा चॅप्टर झाला असेल ह्युमन हेल्थ अँड डिसीजेस, in that chapter there are certain types of immunity are given. For example, immunity means what? Immunity means it is nothing but one of the mechanism of your body which is present inbuilt within your body to fight against certain pathogen or certain microorganism, that is nothing but the immunity. Now that immunity, it is having two types: acquired immunity and innate immunity. Now what is acquired and what is innate? Innate immunity means which is which are present from your birth, that is called as innate immunity. And what is acquired? Acquired immunity, it is nothing but the immunity which we acquire during the life cycle, that is called as acquired immunity. So out of that innate and acquired immunity, these vaccines provide the active acquired immunity. Means, see, there is, shortly I just draw this tree diagram regarding immunity. Okay, this is immunity. Immunity, it is two types: active, sorry, it is innate immunity and acquired and acquired immunity. Okay, innate चे पण दोन types आहेत, that is active and passive. Likewise, acquired, it is also having active and passive. So this vaccine provides active acquired immunity. Okay, so which immunity we are getting from the vaccination? So we are getting active acquired immunity. Okay, so this is the short tree diagram regarding the immunity. Now how this vaccine is formed? So antigenic preparation of microorganism, for example, we are taking the example of snake. Okay, so snake it is having venom, there is a presence of venom. Okay, ते venom जर आपल्याला directly चावला साप, suppose for example, then what happen? आपल्या body मध्ये त्याचं poison होत जाते. So but that venom contains specific protein. If in the laboratory that venom, it is having the presence of protein, and that protein can be incorporated from that venom. Okay, so that protein, it acts as a vaccine to that diseases. For example, कुणाला साप चावला, त्याची वॅक्सिन नाही आहे, बट त्याच्या काही अँटीबॉडीज आहेत, त्या लॅबोरेटरी मध्ये प्रिपेअर करतात, and by the use of that antibody, the person which is suffering from that snake biting. Okay, so that antibody given to that patient, and in this way the patient get cured. Okay, so this is regarding the first example, that how this vaccine prepared and how this microorganism is prepared in laboratory. Okay, so this is regarding the first example. Then second example is hepatitis B vaccines. Okay, so antigenic preparation means what? The antigen causing organism, okay, that we are taking directly into the laboratory, and from that we are making the vaccines. Now usually the vaccine consists of a biological agent. Okay, हे मी तुम्हाला आधीच सांगितलं, vaccine it is nothing but it is having the preparation of biological agent which causing the certain disease. Okay, then this is regarding the, okay, vaccines, it is having also various types. Okay, but for your 12 base only, just you need to remember two types, that is acquired vaccines and attenuated vaccines. Just two types you remember, acquired as well as attenuated, just for general concept. Then vaccines okay, okay, in your book, in your 12th standard book, just one important point regarding vaccine, it is considered, it is oral vaccine. Okay, and which is the best example of this oral vaccine? It is nothing but the polio. It is the oral vaccine na. Other vaccines are directly given on the shoulder. Okay, इथे आमला देतात किंवा इथे forearmला देतात. Okay, so that is nothing but the another types of vaccine. But in your textbook only oral vaccines are given, that is nothing but the polio vaccine. So see, in this lecture we are just stopping this vaccine up to this only, okay? Because how these vaccines are prepared and all, these are very lengthy process. And suppose I started now, but in next lecture it is not connected. So for just short summary, just remember what is vaccines, then what are the examples of vaccine, what are the types of vaccine, how vaccines get functional, okay? But in the next lecture, we are going to study in detail about how this vaccines is prepared in laboratory by using rDNA technology, just like insulin, okay? But we will not stop here right now because the time limit is there. So we are taking the short example regarding the is we are taking the another example of applications, okay? And that another application of biotechnology, it is nothing but it is in the process of agriculture, because within 10 minutes or 15 minutes, within the 10 minutes it get completed. So that's why I'm taking the example of agriculture right now. But in next lecture we are starting the next lecture by the formation of vaccine within the laboratory by using rDNA technology, okay? So in agriculture, see, word itself indicate agriculture. Agriculture means how biotechnology process then carried out by using agriculture. So see, as I already told you, there is one bacteria, soil bacteria, which is mostly used in the agriculture or in the process of biotechnology. That is nothing but the agrobacterium tumefaciens, okay? The bacteria, it is nothing but the agrobacterium tumefaciens. It is the soil growing bacteria. So this bacteria, from this bacteria, we are getting one plasmid, and that is called as Ti plasmid. That is called as Ti plasmid, okay? And these Ti plasmids helps to carry out several processes in the biotechnology, okay? Apart from that, for example, in PTC, that is plant tissue culture techniques. How agriculture application is carried out in the agriculture in PTC. PTC means it is plant tissue culture. What exactly happened in the process of plant tissue culture? In the process of plant tissue culture, we are preparing the plants within the laboratory, and from certain growth, that laboratory plant get incorporated into the soil. Now, what exactly happened in the PTC? So, see, for example, see, there are several terminologies. For example, explant, then callus, okay? Apart from callus, then organogenesis, okay? After organogenesis, one term is also given in your textbook, that is micropropagation. So what is the meaning of this micropropagation? Micropropagation means it is the technique where we can form multiple plants within a very short time. That is nothing but the process of micropropagation, okay? And this micropropagation process is carried out by means of PTC, that is plant tissue culture. What happened in this plant tissue culture? For example, suppose this is plant, this is root, this is stem, and this is, for example, leaf, okay? For example, this is leaf, and for example, this is flower. So what happened in plant tissue culture? We are getting the part of plant. Now, which part? For example, we are getting only the stem portion. We are getting for root portion. We are getting the leaflet portion, okay? Or we are getting the flower portion, okay? So what is explant then? Explant means the part of the plant which we are getting from the plant, that is called as explant. It may be stem, it may be root, it may be leaves, okay? Then what happened? See, within the laboratory there is a presence of test tube, okay? Within that test tube there is already the preparation of media is done, okay? For example, Which media is used in PTC? जर क्वेश्चन आला MCQ मध्ये नीट मध्ये स्पेशली, then it is MS Murashik and Skoog. Okay, MS media is most widely used in plant tissue culture. That is Murashik and Skoog media. So these media and along with some nutrient, for example carbon containing, then certain growth hormone is also given within this test tube. Okay, so on this media the explant which is given, it is incorporated on this media and it takes for several days. After 8 days these explant is grown into the broad circular or abundant growth like structure, and that is nothing but the callus. That is nothing but the callus. So what is callus? Callus, it is nothing but the undifferentiated forms of the growth from the explant, that is called as callus. Okay, so from explant there is a formation of callus, then from that callus there is a synthesis of initial synthesis of root and stem. Okay, then after and our initial synthesis of root and stem, it is nothing but the process of organogenesis. Okay, and then after that, from several days, means within a 2 week, we can show within this test tube there is a formation of plant which is having leaf, root, and stem. So this is nothing but the all process of plant tissue culture which is carried out in the biotechnology, that is plant tissue culture technology. It is the one of the technique of biotechnology, that is plant tissue culture, and in this way we can form multiple plants. Okay, we can form multiple plant within the laboratory, and after that this plant is incorporated into the normal soil, okay, for further growth. But after callus formation, okay, this process is carried out in well defined chamber, means in laboratory there is a presence of specific chamber. Within that chamber proper sunlight, proper food, proper water, proper nutrients all are provided by just one setting. Okay, so this is regarding the production of plant, means high amount of plant by the use of biotechnology process. Then the second which is given in your table, first it is the micropropagation, and second it is there are several plants which are infected by certain diseases. For example, cotton. Okay, cotton, it is nothing but one of the crop which is widely form in the farm. Okay, so what happened, see, on that cotton there is a formation of yellowish fungus like structure, and that is nothing but one of the disease which is caused by that plant. So that diseases, or there are several diseases which are formed towards the plant to avoid all that diseases, and within a short time to get a pure and very good product of plant, there is a terminology, that is plant tissue culture is there. So what happened, see, तुम्ही बीटी कॉटन एक example ऐकलं असेल भरपूरदा. BT cotton, it is nothing but one of the strain of cotton, that is Bacillus thuringiensis cotton. Okay, so what happened, the diseases, okay, that which is occurred on the cotton as well as other plants, that get vanish. Okay, that gets vanished by this species, that is nothing but the BT cotton, and BT means it is Bacillus thuringiensis. It is one of the Bacillus thuringiensis. BT means it is Bacillus thuringiensis. It is nothing but one of the microorganism or organisms, okay, which is capable to remove all. So this is regarding the application of biotechnology in the process of agriculture. So see, there are certain examples given in your textbook, that is nothing but BT cotton, then pest resistant plants. Okay, ओके, असंच सांगितलंय सगळं जे मी तुम्हाला शिकवलं आता, प्लांट टिश्यू कल्चर अँड क्लोन. ओके, वन एक्झाम्पल इज गिवन, दॅट इज नथिंग बट क्लोन. Now what is the clone? Clone means it is nothing but the preparation of multiple copies or exact and multiple copies of organism, that is called as clone. So what happened in the process of PTC? In PTC, we are preparing the plant which is having specific character. Now, which specific character? The plant, it is capable to fight against all the diseases. For example, is to fight against yeast, to fight against pests, to fight against weeds. Okay, so that are nothing but the characteristics of plant tissue culture made up of plant. Okay, means plant जे plant tissue culture technique पासन grow झालेले आहे, or the plant which are produced by using PTC techniques, okay, that is having the capability to fight against all the diseases which plants can survive. This is regarding the PTC. And clone, it is nothing but it is the formation of exact and similar sequences of character. That is nothing but the clone. So what happened in agriculture? There is a formation of clones of various strains of species, okay, to avoid that diseases condition, as well as to produce or to increase the agricultural product, okay, with maximum fighting strength. That is nothing but the application of biotechnology in agriculture. Now the next application is given in the gene therapy. That is nothing but the third gene therapy. So what happened in that gene therapy, that we are going to study in next lecture, because as I already told you, I stopped that vaccine steps. Okay, so that vaccine step, that is the formation of vaccines by using rDNA technology within that laboratory, it is somehow connected to this gene therapy. Okay, so that's why I'm saying in next lecture we are going to study this gene therapy along with that pending vaccines preparation steps along with rDNA technology. So this is regarding your two applications, that is application of biotechnology in medicine as well as application of biotechnology in the agriculture field. Now there is word one only given, that is recalcitrant. Recalcitrant. Okay. So this word, one word is mentioned in your textbook. Okay, in the agriculture. So what is the meaning of this recalcitrant? So recalcitrant, it is nothing but the reduction in the soil seed moisture content only. Okay, so what is that recalcitrant? Recalcitrant means it is the reduction in the seed content. Some seeds are so moist, okay, so we can preserve for a long period. So by using PTC technique, we are trying to incorporate or to make the seeds having genetic constitution that are capable to having the low moist content, so that can be preservable for a longer period. So that is nothing but one of the important term which is mentioned in your textbook. So this is regarding our today's lecture. Hope you all understood very well regarding the applications of biotechnology. In the next lecture, we are going to study gene therapy as well as the several steps which are remaining during vaccine production. So till then, bye.